Next speaker is Dr. Hidewaki Nakagawa, Team Leader, Riken Center for Integrative Medicine Science, Laboratory for Cancer Genomes, Japan. Time is short, so please read his career on the textbook. His talk is Whole Genome and Immune Genome Landscape of Liver Cancer. Dr. Hidewaki Nakagawa, please.
So thank you very much for inviting me, and also thank you for the committee members and organizers, and also the professor Tanaka. So I'm just talking about the whole genome sequencing for the liver cancers, and this is kind of one of the project of the ICG TCG project on world. So since 2009, we have a sequencing of the whole genome of 300 of the liver cancer in Japan, and the most of the HCC and also the half of the HCV, and in the quarter is the B virus and the non-B non-C virus. The data is basically it's a huge. We have also the sequencing RNA, and sequencing of tumor DNA and the blood DNA, and totally about 70 terabase data. It's basically 300 terabyte data. So we need of the supercomputer systems. We collaborated with the Tokyo University Shogun supercomputer system, and we sequencing of this kind of 300 whole genome sequencing of liver cancer. So this is a result of the somatic mutation numbers of each of the liver cancer. The median number of around the 10,000, around the 10,000 mutation in the whole genome level, but the number of the very variety of various. And we have it's 10 times more of somatic mutation samples here. This is obviously showing the MSI positive of liver cancer. That's only one sample. So among this kind of data, we try to find driver genes of driver mutation, basically of the significantly frequently mutated genes. It's coding genes. P53 is the beta-catenin, ARID2, ARID1, and action APCs. We have several kind of many of the driver genes. We call the long tails because we have more data. We have more and more driver genes. And also this is a whole genome sequencing. We have tried to find out the non-coding mutations, but only one. This is a very the frequently mutated is the TAT promoter mutations, and also we have some amount of the significantly mutated the non-coding genes like MARAT1A, MARAT-B, and also this is a non-coding NIT1. This is also linked RNAs. And also that we have some amount of promoter mutations, but this is minor. And also that the whole genome sequencing can be detected the structure variations. One sample is a tremendous number of the structure variations. One sample is a kind of shows in the chromosynthesis. And this is a list of the structure variation affected genes. TART, including TART and APC, cyclin D1, P60s, and some amount of the famous the driver tumor suppressor genes. Among them, it's a number one genes, the driver genes in the frequently mutated genes is TART in the liver cancer. The half of the liver cancer have shown promoter point mutations. And also we detected the structure variations of promoter regions, and also the B virus integration in the TAT promoter regions. And this structure variation is related with TAT overexpressions. So basically this is the small ranking in the liver cancer driver genes. It's Yokozuna basically is a Wnt signal pathway, including beta-catenin action APCs, and also the TART. The OZK is a P53s, and also we have other chromatin regulators, including ARID2, ARID1, BRAT7, and also the many long tails of the driver genes as mutated in the liver cancer. So among these of the driver genes mutation data, and we have tried to the more systematic analysis, and by using of the protein-protein interactions and also the classifiers of the mutated genes, and also the classifiers of the liver cancer, molecular classification of liver cancers. Basically, we have 5 or 6 on the class like this, and ARID1 prominent, the P50C prominent, P60 prominent of the liver cancers. And it is interesting about P53 groups and also ARID2 groups it's a worse prognosis on our cohort. So this is the point mutations and also structure variations on the liver cancer genome. And also whole genome sequencing can be detected of B virus in other virus. And we detected obviously it's we have a sequencing of B virus of liver cancers, and we have tons of the B virus sequencing leads. But 9 samples have this negative, show the negative is the serological test of a B virus. But the sequencing analysis detected a number of many of B virus leads, sequence leads, and also some sample is integrated of human genome. And also this whole genome sequencing can be detected the pinpoint of the integration site of the human genome. We have detected in the whole genome sequencing can be detected in the more than 200 of integration site of B virus. It's one of the preferential site of the TAT on the TAT promoter regions, and ML4 on the SOX5. Otherwise, the integration is a singleton of integration site. So the next, we try to characterize on the more of B virus integrations. We have the sequencing, ultra deep sequencing after capturing B virus sequence lead from 100 of B virus infected liver tissues. We detected more than the 1,600 of integration site of human tissues. Liver tumors and non-liver hepatitis tissues, and basically is detected of the preferential site of in the non-liver, non-tumor site of the fibronectins, and the liver and the tumor site is the TAT on the ML4, and which is consistent in the previous data. Basically is B virus integration can be occur preferentially in the genomic region of basically is whose chromatin structure is open or highly expressed, and this kind of site is affected, frequently affected by B virus integrations. And also that we have the using of the mouse model in the B virus infection models. The chimeric, human chimeric mouse, we infected with B virus, and also the follow-up and and also the obtained of the liver tissues after the infections of day 10, then 20, 3 and 4 and weeks, and the sequencing of after capturing of B virus sequencing, and with sequencing of ultra deep sequencing with B virus. And interestingly, we found it's after 7 weeks, we found it's some amount of chimeric leads, which means suggested of the B virus integration, human genome. And interestingly, the B virus integration occur preferentially in a human mitochondrial genome around 4 and 5 weeks after B virus infection in these models. And also the B virus, the integration site, also it's a very preferential site is here. So this is, but unfortunately, this is a kind of a models on infections. In the human samples, we don't have detected the B virus integration, the mitochondrial integrations of a human sample. And this is also that we try to characterize another virus. We have de novo assemble of RNA sequencing data and making contact and to search over the database of other virus. So top three is basically the AB virus. We detected the AB virus in it's a 4 and 5 samples. It's like this. So this is the AB lead in the human liver tissues, and some of 3 and 4 samples have tremendous number of AB virus lead. And among them, the 3 samples is by ABB virus in the integrated of a human genome. So the next one is we try to understanding of mutation signatures, which have basically we have detected 10,000 or 7,000, several thousand of mutations in the whole genome. The mathematical analysis, it's the analyzing of the convoluted of somatic mutation like this. So basically, it's in 5 or 6 in the substitution patterns, and the mathematical analysis in the classifies of mutation 1, 2, 3, and 4, and 5, 6, 7, many. Now the international community is like 30 and 40 mutation signature. And basically, the liver cancer is one of the most known about its etiologies. So we found it's basically is the signature 1. This is reported related to the aging, and liver cancer is basically in aflatoxin. We have detected aflatoxin signatures, signature 24, and also the aristolatic acid signatures. This is very rare in the Japanese liver cancer, but it's very common in the Chinese liver cancers. And also that we detected the smoking signatures, alcohol signatures, and also the liver cancer specific unknown etiological signature like this. And also that by using of such kind of signature analysis, we try to estimate the origin of the liver cancers. Basically, the liver cancer is classified the HCC, ICC, and also the combined type like this. Especially for the combined type, basically think about this is the same origins of the 2 phenotype. So we have sequencing of ICC of the HCCs and try to compare their signatures with the hepatitis background. Interestingly, the HCC and the ICCs with an hepatitis background is very similar signatures. And also we have collaborating on these groups, and also these groups is obtains some good algorithm. It's for the estimated cellular origins. Basically, the somatic mutation distribution in the whole genome is basically correlated with an epigenetic or chromatin mark in a specific tissues or cells, like liver tissues or stomach tissues or small intestines or brain or other many. We have tons of the data of some specific chromatin mark. So this is the data for the melanoma, these chromatin structures, the epigenetic mark, and of the mutation distribution. It's very, very similar like this. By using this information, we have established a good algorithm to estimate the cellular origins, and obtain try to estimate the cellular origin of liver cancers. So the ICGC tissue data estimated that most of the liver cancer HCCs obviously is originated from the hepatocyte. That means it's very similar of signatures. But when we focused on the ICCs, especially for the ICC within the hepatitis or virus infected ICCs, it's more than half of the ICCs within virus infections. It should be the estimated their origin is the hepatocyte. And also the HCC, it's not all HCC is not estimated to be originated from the liver cancer hepatocyte. And this sample is basically enriched of the EPCAM of cytokeratin positive of HCCs. So let's talk about immunosignatures. So basically we have also tons of the RNA sequence data. and with liver cancer and also the background hepatitis. And we have, this is the kind of the estimated of the inflammations by using information granzyme perforin expressions, which is the cytolytic activities in the tissues. By using information, we have two groups in the very hot groups and also the cold groups. The overall survival is in hot groups. It's inflamed of liver cancer is very good. On the other hand, the cold one is not good. On the other hand, it's a non-liver tissues, hepatitis tissues. The cold tumor, cold liver is it's a better prognosis. And on the other hand, it's a highly inflamed of our liver tissue is a bad prognosis. And also that we have using of some mutational signature data of RN6, so we have estimated T-cell inflamed signatures, and a highly T-cell inflamed signature is a good prognosis. So we try to the expanding of this kind of strategies by using of the many signature analysis, immunosignature analysis, using informations like the such kind of the immunospecific genes analysis, and compare to the inflammations of a tumor and of the hepatitis livers. You can see here, it's a red means it's kind of hot inflammations. It's cold inflammations. You can see here, it is the same patients of liver and tumors. In the liver tumor, it's more hot. Liver tumor is less and less inflamed than the liver tumor, non-tumors hepatitis livers. That means we estimate it's probably many of immunosuppressive mechanism can be working in the liver tumor. So we try to classify the liver tumors based on the immunosuppression mechanism, and by using such kind of immunosignatures. And finally, we have 4 clusters of immunosuppression mechanism in liver tumors. One is beta-catenin signatures, one is Treg signatures, and this is also the TAM signatures, macrophage signatures, and also these immunosuppressive negative signatures like this. And this is significantly related with Chan's subclass of a molecular subclass of liver cancers. Proliferation subtype is related with the T cell, dominant of the C cell functions. And the polysome 7 subclass is showing the dominant of the TAM signatures. Wnt signal pathways, beta-catenin subtype is obviously it's beta-catenin signatures. And also the interferon subtype of molecular subtypes shows the immunosuppression deficient subtype. And also that we have tried to the association of mutations of such kind of immunosignatures of liver cancers. Obviously, it's beta-catenin signature is beta-catenin mutations. It's very strongly, significantly strongly of the exhaustion, T cell signation, also Treg signatures of such kind of immunosignatures. And also the interesting of ARID2 mutations, including of structure variations of other copy number changing. This is also related to the macrophage M2 signatures. So the Professor Tanaka in the Tokyo Medical Dental University is a provider of the data of the ARID2 knockout HC cell line data, and we compared of the expression profiles. And the one with the knockout of ARID2s, this is knockout and the wild type, and there are some amount of cytokine or chemokine expression is changing significantly ARID2 in the cell line. So that means probably ARID2 mutation is related with the immunosignature of liver cancer. So this is some of these. We have a sequencing a whole genome of liver cancer, try to identify the driver of genes, mutations, coding, non-coding, virus infections, virus integrations, mutation signatures and etiologies, and also try to estimate the cell origin of a viral phenotype. And also the immunogenomic analysis is be classified of the 4 groups. Basically, it's immunosuppressive 4 groups, and TDAC, beta-catenin, TAM, and immunosuppressive deficient subtype. And also some somatic mutations, including beta-catenin ARID2, is related with an immunomicroenvironment liver cancers. This is contributions. The Dr. Fujimoto is now in Kyoto universities in our groups. Then now is moved to the Kyoto universities. He is working on the tremendous work in the whole genome sequencing data analysis. And Dr. Fujita is also the working immunogenomic analysis in our data. And also that we want to thank you for, especially for some clinicians, which we provide us and a very valuable sample, liver cancer sample of tissues. Thank you for attention.
Thank you, Dr. Nakagawa. Could you please accept several questions from floor? Sure. Is there any question? Floor? No? Dr. Nishida, please.
Thank you for the wonderful talk. So I'm the Kindai from the Kindai University. And based on this analysis, some metabolic genes, such as albumin, is always listed at the top of the mutated gene, but generally it is not considered that this gene is a driver gene. So what this happens?
Yes, we have a long discussion about the albumin mutation in liver cancers within other collaborators and also the international communities of cancer genome. Basically, we believe albumin is not driver genes, and basically albumin is one of the highly expressed genes in the human bodies, and such kind of a gene is always in the open, showing the open chromatins, and be very vulnerable for the other genomic stress and other mutated accumulated. So we believe that albumin is just like we were just like showing, watching this open chromatin structure of albumin genes, albumin lesions.
But actually, it is the mutational type of the albumin gene is induce a clonal expansion. I mean, it's every mutation is the same in the clone, among the clone, right? But actually, it is not affected by the driver gene, right?
No, it's not that.
Okay, so thank you.
Thank you very much, Professor Nakagawa. Thank you. Oh, Professor Omata, please.
The whole landscape almost you can imagine, but the thing is missing is that, and then we are hoping too, is that NASH, because this is a very mysterious one for us. Integration, yes, maybe reported Southern blot in 1979 already.
Yes.
And then you carefully check all the things. But nothing, no clue at all about the NASH. So for the next maybe 20, 30 years, nobody can get the answers. The question is, all those patients are getting the cancer without background fibrosis, many of them. That's not found in many of the cases, especially due to C. So that's one mystery. And then, so, I mean, we need somebody. We need a help like you to take me or take us to the new paradigms by these methods. So, and then according to your signature, I didn't see anything about the NASH.
Yes, we have, we're sequencing of 300, and among them, there are some samples. It's probably showing the NASH-oriented one, and especially for the non-B virus one, and we don't have any. Some samples don't have any B virus sequencing lead, not occult B virus, and likely to be a NASH. and also there is no drink alcohol at all, and NASH. And we just like compared our signatures and other stuff, and not so different from the other, for example, C virus one in a genome.
Probably pretty soon, from June, the Foundation One or some of the company gonna get started about the panel sequencing, and then can find out some possible druggable promoter mutations. And in fact, we are doing the sequencing for the last 5 years, more than 2,000 cases by using the panels, which is spanning about 260,000. So we are getting pretty much an idea that what can be the actionable targets. But the key is, for example, you said Grand Champion or Ozeki, the TP53, no drugs,
Yes.
KRAS the same. So I think to me, it looks like the actionable mutation is not that great. But not that great is not the great of the number of drugs. So that's, not your fault and not our fault, but company is not catching up at the speed you found new actionable mutations. So that's this situation, and then that's a dilemma of our clinicians.
So we also evaluated actionability of such kind of 300 samples, including about copy number changing, about structure variations. So the Mungzen, I think the 40% of our liver cancer in our sample is likely to be the actionable, possible actionable, including, especially for the copy number changing is probably important to, to think about actionability of liver cancer in other type of cancers. PTM loss is obviously, it can be some frequently detected of liver cancers, and this is a kind of a very good target. Also the, other type of, amplifications is also the detected of, not so frequently of, one sample, two sample, or HER2, other stuff. And so I think it's the clinically, as the copy number changing, it should be examined in the, such kind of clinical sequencing method, basically.
Very simple question. Herceptin is targeting the amplification, or just Herceptin is?
Yes.
So that's a typical ones, right? All right. Thank you very much.
Thank you.